From SigOpt, an Intel company, I'm Mike McCourt, and this is Experiment Exchange.
How do you extract health data? How do you share health data? And how do you ensure user privacy?
Today, we have with us Alexander Johansen, Ph.D. student in computer science at Stanford. His work focuses on how deep learning can play an increasingly significant role in the future of medicine. We will start by discussing how SigOpt was a key tool in maximizing the performance of his neural networks, which were originally developed for natural language processing, but are now being applied to proteomics. We also discuss how new advances in medicine and therapeutics can be found through machine learning, but that unique challenges exist in these pursuits, such as access to medical data and the need for privacy ensuring methods. Ladies and gentlemen, thank you for joining us again on another one of our interviews. In particular, today we have Alexander Johansen, PhD student in computer science at Stanford, joining us. Alexander, thank you so much for coming today.
Thank you for having me.
Absolutely. I'd like for a moment to talk about both what you're talking about at our SigOpt Summit. For those of you who don't know, that's summit.sigopt.com. But I'd also like people to take an opportunity to learn more about you. So why don't you tell us something about yourself, please?
Yeah, so I'm a second-year PhD student at Stanford University, and I am studying the intersection between computer science, bioinformatics, and digital health, and how we might use computer science to answer important questions such as how do you extract health data, how do you share health data, and how do you ensure user privacy?
That's a big topic right there. There's a lot of stuff going on in that particular topic. Let's kick it off right there. Let's think about that. Tell me about your lab. Tell me about the kind of things you work on, how you try and approach this very complicated intersection of various things.
Yeah, so I think a lot of people today are starting to ask questions about health and about user data. And so we've seen in the last 100 years, if you look through anthropological and even archaeological studies, that the human body and even the human skeletal structure has changed vastly during the last century. And we don't really know why. We don't necessarily know whether or not this is caused by lifestyle changes or any other different types of changes, and where we might go make changes to the way that we live, the way that we handle pregnancies, the way that we handle childhood, and the way that we handle treatment of chronic illnesses. And so in here, there's a new exciting field of research called digital health that came about some 6-7 years ago. And digital health, which a lot of you might be familiar with the Apple Watch and the Fitbits of this world, but it's the idea that we can sort of continuously measure in a distributed way, continuously measure also for cheap money, humans on a vast scale. And now with this comes a lot of questions, such as what's the best device to use? Once you have this device or any type of, for example, a 23andMe genetics test, how might you be able to extract this data, get access to this data? What about usage rights of this data? Does 23andMe own your data? Does Fitbit own your data? If you want to build any type of medical algorithms on it or get medical insight to it, that's very important. And also, let's say that you want to use this device for some type of clinical study. You want to get insight into diabetes. You want to get insight into pregnancies, miscarriages. Could be an example of a clinical study. How might you ensure that the people who use the Fitbits throughout these pregnancies, that they can delete their data once they're done with the study, that it's not owned by Fitbit, that it's privacy insured? And we're also seeing a lot of hackings that happens all the time for all of these different wearables companies. Do we have de-identified data? Do they have secure connections? So there's a whole array of privacy questions security questions that we think, especially with the current atmosphere around data privacy, which we've seen with a lot of the big tech giants, that we ask these questions right here, like in an academic sense, and we sort of highlight the issues around it, such that we can come out with solutions that solve these issues. So, but right now we're mainly focused on figuring out how might we be able to find this combination of devices, and how might we be able to handle some of these privacy concerns.
I see. And in reality, then the problem isn't just one of getting data, using data, but there's also, as you're talking about, ethical concerns, practical concerns, security concerns. Even if people agree that somehow there is no ethical quandary, you still have to be worried about people stealing it, because I can't change my fingerprints, can't change my blood type, can't change any of this. So if somebody gets that info, it's there forever. Do you have people in your lab who are philosophers, or what kind of person deals with these questions?
So we actually just ran a competition for volunteers to join the lab. So we do most of our recruiting on Reddit. well, we because I think there's a lot of talent out there today who do not have the opportunities to work together with exciting research lab on exciting research projects. So we find a lot. We have about, I think, 8 people, maybe 9 people today. And a lot of the talent we actually get from people from abroad who might not be in the vicinity of a big research institution. And then we go out and then we ask them these fundamental questions. So we try to get computer scientists, biomedical engineers, and so forth, who has mainly been trained in sort of how do you create things? How do you make databases, the classical core computer science courses, which unfortunately do not deal with data privacy and ethics at all. And then for the competitions that we put out for them, we ask them these questions, because we want people who are well-rounded and who are able to think about that, who are able to see sort of the societal problems. And in the review paper that we are constructing right now, we're going to make a historical view on not just data privacy, but also questions within medical grading, which I think is very interesting nowadays, especially when you see all these different vaccine trials, right? Like COVID, Pfizer vaccines, Moderna vaccines, and so forth. What does that actually mean? Like these different FDA approvals, these different FDA processes. What can you expect from them? What about transparency? What about you as a user actually understanding all of these processes? And so we want to highlight sort of the history behind these different legal documents. And we also want to highlight what it means that something is FDA cleared. What does it mean something is FDA registered, FDA approved? What does it mean a soft piece of software is FDA approved? And what stages are the FDA approved for? And what can you trust this device to be when it has these different levels of approvals? So those are also some of the things that we are very interested in right now and that we're looking into for our recent paper.
That's a sort of a big topic, and I love to hear that this is being addressed, this is being considered right now. One question that sort of pops up as I'm hearing you talk about this, the idea that the FDA, in particular, primarily in this case, the U.S. then FDA, is the one that's in charge of this. Obviously, there's an element of it, especially as we talk about drugs and therapeutics and treatment, where that makes logical sense. Are there other government entities that somehow are most logically meant to be involved or should be involved in this discussion? Or do you think that the FDA will be the entity where a lot of this discussion takes place?
That is a good question. I think right now, there is not a lot of laws around wearable data. So unless you are actively using it for treatment, which means that it's sort of like it has to fit inside of this treatment space, which is it's something you can apply for your insurance to get. You can apply for your insurance to get like a specific device, a pacemaker. I mean, that's effectively a wearable, right?
Absolutely.
You can apply to get a pacemaker, you get a surgery, you get it put in, and all of this is through your insurance. You go by a hospital, you have treatments, and so as long as it's sort of a part of your medical treatment, then there's a set of rules around that. Like for example, it needs to comply with HIPAA. Um, but for a lot of these wearable devices, they're kind of seen as sports devices, which means that there's not a lot of legal protection around it. There's not a lot of legal protections for the user.
Mm-hmm.
And the amount of data and information that you can gather for the user is extremely valuable and is can be used for treatment and can be integrated into your personal treatment. But we could also see a future where if we don't have strong privacy regulations for this, then a lot of that data might also be utilized by companies that you might not want to utilize that data. So a recent study found that Oura rings can predict menstrual cycles. And so by that, you might even be able to predict pregnancies, right? Like if you're off your menstrual cycle or menopauses and so forth. And that information might not be something that you want shared with everybody. And so, given that there's currently not a lot of restrictions around what you use the wearable data for, that might lead to some privacy issues down the
Mm-hmm.
road. Um,
Mm-hmm.
and
Yeah,
and
it's a
yeah,
great point.
and so right now it's only if it's used for medical purposes and intents that the FDA is involved. Other than that, there's not much involvement from any government entity.
Very interesting. Very interesting, and it does feel like something that more discussion, more thought will need to take place on. Action will need to take place at some point. And I love to hear that outstanding researchers at Stanford right now are trying to be at the forefront and really thinking about what the implications are before it's a little too late, before we lack the ability to take proactive action. I'd like to talk a little bit now about the particular project that you spoke on at the summit. The title of your talk, Deep Learning for Proteomics and the Future of Medicine. Again, very ambitious, very exciting here. Maybe you could break down a little bit of what you spoke on in your talk.
Yeah, so we have two different research directions at our lab. One of them is more basic research within bioinformatics. And so here we've seen that contemporary deep learning tools can be used for handling proteomics challenges. So
Mm-hmm.
if you actually look at a protein is very similar to a string of text or a paragraph of text. It consists of amino acids, and these amino acids have a vocabulary of about 20 different characters, which is very similar to the English language, right?
Mm-hmm.
Of 27. And these different characters will be strung together in a sequence of about, on average, about 300 characters,
I
and
see.
which is quite similar to a paragraph in length.
Yeah, yeah, I can see that.
And because of this similarity, and there's also a lot of other similar, like for example, there's structure. So there's structure in these sequences because they actually have a physical meaning. Like they fold in a specific way,
Hmm.
and they will attach to other proteins, and you can target specific receptor on these proteins. Like they will fold in specific ways that will allow drugs, chemicals, and so forth to attach to them and change.
I see.
And because of that, researchers start thinking that, okay, well, might we be able to use some of the same methodological work that we've been using for natural language processing on proteins?
I
And
can
so
see that
this,
makes sense.
recently, in the past 3 years or so, there's been this move towards using these big semi-supervised or unsupervised language models in order to generate interesting embeddings for text.
Mm-hmm.
And so about a year and a half ago, researchers started asking the same question. Well, can we do this for proteins? And it turns out, yes, we can. Um,
Cool. Very
uh,
cool.
and so we have been collaborating with the Technical University of Denmark, who has some of the world's leading tools for protein predictions. So that's predicting a whole range of functions for proteins, which is important in every single aspect of life sciences. So for medicine, for plant technology, for bacteria and so forth. Like if you want to have bacteria that like breaks down plastic, well, then they need to act in certain ways, and we need to be able to predict how they act, and whether or not we can design specific proteins and enzymes that can help us solve the task that we're interested in.
I see.
And so we started thinking like, okay, can we use these protein language models to solve those problems? And it turns out, yes, we can. and it works a lot better than what we've been dealing with previously, because it's able to learn the underlying structure of proteins and how proteins relate to each other. And it's able to learn not just the different types of functionalities, even the functionalities that we're looking to predict. It has learned this just by looking at a whole range of proteins.
Interesting.
And so this it will be predicting signal peptides, secondary structures, whether or not it's soluble, whether or not you can express specific enzymes in a bacteria. So on a whole range of protein tasks, we've seen that this has been very useful. And not just that, but often, and especially for basic sciences like proteomics, we have the issue that getting label data is very expensive.
I believe it. Absolutely.
And so a lot of these data sets will only have hundreds, maybe just a few thousands of labeled proteins. And these would have been gathered by the whole community through the last maybe 20, 30 years.
Long time. Yeah, right.
Exactly.
So in this case, you have the ability to sort of, is this generate? Is this like a simulated data situation?
So it's more in the sense that we might, for all of this, so we have big databases of like hundreds of millions of proteins, but we might not have any label for these proteins. So now what these language models are able to do is that they're able to look through these large databases. They're able to understand fundamentally, okay, what goes on in these proteins, what sort of differ these proteins from these proteins.
I see.
Because it always wants to be able to predict the remainder of the protein, so it needs to understand what is going on, sort of like what is the core functionality of this protein.
I see.
And what we do is that we then go in and say like, oh, like this subsection of proteins, well, they express this specific behavior. And then these language models are able to, because of sort of the context vector that they create, they go like, oh, well, if these express in this way, then the rest of these are probably also expressing this way.
Gotcha.
And so we found that it's extremely well suited to handle proteins that are very distant from what we're training on. So it generalizes really well.
It's incredible.
And for this, we in order to optimize this, we actually use SigOpt. So we're dealing with these large neural networks, and we need to optimize learning rates, how we handle the output from these neural networks, hyperparameter optimization, and so forth. And a lot of this can be quite cumbersome, and it's also can be very computationally expensive, because fine-tuning these big language models might have, I think the one we used for our latest research had 650 million parameters.
Wow.
And so this takes a lot of computational resources, and because of that, we figured, hey, well, let's use SigOpt in order to see if we can grind out performances. And so that has just, I think it's kind of a mixture of giving us a little bit of an edge, combined with also just the simplicity. Like we don't have to think about it. We just pipe it into SigOpt, and SigOpt just does the hyperparameter sweep for us. And it probably does a better search than we would do, where we would just like do like a grid search or something along those lines. And also definitely also the explorative part of it, which so we've also used it previously when we try to build our own language models. And so especially for some of these more sort of like foreign style, like archaea proteins, we found it, and virus proteins, we found it very difficult to build language model for those. But so we try to just allow SigOpt to just do full exploration around how do you put together a language model for specific proteins for virus and archaea. And then we just let it run, I think, for like 200 or 300 iterations, just exploring the space of how might you be able to build a language model for this. And we found for these, I think it ran 120 iterations where it was just having complete random perplexity. And then all of a sudden, it just caught it. It just found like the right set of hyperparameters, and we were able to, I think we dropped to perplexity of about, so we dropped from like 18 to about half of that, about like 9 or 10 or something. So
Wow.
it went from being able to be completely random, we weren't able to capture any type of information about this, to explore enough until it actually found a suitable combination of hyperparameters that could solve the problem. So for this like more constrained problem of like fine-tune the language model, it sort of gives us convenience and grind out a few more percentages. And for these more sort of open new avenue problems, such as like building language models for very unique type of protein families, it helps us a lot with exploration as well.
I believe it. And that's just so exciting to hear. It's so exciting to hear that the tool's providing value and that it's easy to use for you. I have both a topical question and then a personal question. Let me start with the personal one. You've spoken now at great depth about the actual impact of what you're doing with precision on the proteomics community and the research that's being done there. How much have you had to immerse yourself in that topic and learn about proteomics in order to be able to read that research and then contribute back to it?
That's a good question. That's I think that I'm a trained computer scientist. Um, And I think that in computer science, you're very much focused on the computer science aspect of things. Like, how can you build these scalable algorithms? How can you work with mathematics? How can you deploy this to web service and so forth? And you barely get exposed to applications at all.
Mm-hmm.
I think this is a key issue. And I think that we should definitely have a bit more of an integrative approach to things, which is also why a lot of the wearables projects that we are working on, we want to be able to make this useful for people who are exploring sort of the intersection between computer science and health. We want to be able to make this used by people who might come from a straight computer science background. Now, from in the case of me, I found bioinformaticians, and then I spoke with them, and then I took the time to delve into what are the problems that they are facing,
Mm-hmm.
and I was able to extrapolate those problems to sort of key math equations and key math problems,
Mm-hmm.
and then from those abstractions, it was just a computer science problem,
Gotcha.
and that didn't really change throughout the process of the algorithmic development, or the process of writing the paper.
Mm-hmm.
The only thing that is a challenge though, and so I think this mainly calls for close collaborations, is that while the CS computer science community is mainly interested in algorithmic development, the life science community is much more interested in biological interpretations.
Yep. Yep.
So beyond just being able to have a better algorithm, we also need to give some biological interpretations, like why is it important that we get a better performance on this specific task?
Gotcha.
What biological insight might we be able to get from this new tool? And so for this, it has been key that we have collaborated with, in this case, the Technical University of Denmark. Henrik Nielsen, who is a associate professor at that institution.
Excellent. I love to hear it. And Dr. Nelson, thank you for being part of this exciting collaboration. The other element I wanted to talk about briefly was earlier on, you had talked about this idea of the more data we get from the more sources, the more you're going to be able to do exciting things like this. Now, this isn't unique to proteomics anymore. Now, this is more about this broader wellness discussion. We have the Fitbits, which we take our blood pressure, maybe any number of different things. All of these help us get more data. But then at the same time, the question is, what piece of data is somehow most useful or most valuable, most pertinent in putting together this picture of wellness? In my mind, when I hear you talk about that, that feels a lot like this question of explore exploit balance. How do I pick the right piece of data to learn more about the person, but also the most valuable piece of data so as to best shape their wellness outcomes, whether it's lower their blood pressure, lower their cholesterol, whatever number of things it is. How is that research proceeding right now?
So kind of like the balance between what data is most valuable.
Mm-hmm.
Yeah, so that's very much an optimization problem. And I think a lot of this also comes down to cost,
Mm,
because a lot of these. So, for example, a Fitbit is not that expensive. I think it's $80 or $120 or something along those lines. But let's say that you want to do a, single cell RNA sequencing of a specific tissue or some blood. Well, that can run you $4,000.
a lot. Yeah.
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 So being able to understand what piece of information amongst this wide variety of data inputs that we have might be most useful. So let's say
Gotcha.
that you can choose between 100 different tests and 100 different wearable devices.
Hmm.
Um, and they all have some type of cost. They all cost some amount of money.
Hmm.
When should you use what?
Yeah.
Should I 2 weeks from now get single cell sequencing, or should I get a, blood panel first and then run some Fitbits and maybe some EG headbands,
Hmm.
and then based on that, maybe I don't need single cell sequencing, maybe I should get single cell sequencing. So we definitely need to have some type of balance in between exploration and exploitation.
Gotcha.
And I think that this could be extraordinarily valuable, for any type of clinical study or clinical trial,
Absolutely.
because a lot of the big grants given out by the NIH today are mainly for lab expenses, such
Mm.
as, single cell sequencing is probably one of the biggest ones right now,
Hmm.
or gene sequencing,
Okay.
because you will end up spending a lot of money on that. Um,
Gotcha.
so being able to optimize where do we have information gradient, will be, a key point of research going forward.
That makes perfect sense. And that is a point of research which I will be very excited to see the outcome of, and potentially assist in the outcome of, as we have the opportunity to continue supporting SigOpt out in the world. Alexander, thank you so much for talking to us today. Where can people go to learn more about you and your research?
Yes, so, our lab is, you can find our lab website at stanford-health.github.io. Or you can go directly to my Twitter handle at AlexRoseJoe, and see the different tweets that we have on these topics.
Very cool. Very exciting. Thank you very much for joining us. Thank you, everybody, for watching. We hope to see you next time. Take care. Have a wonderful day.
Thank you.
If you have enjoyed these interviews and want to learn more, the description below contains the link to the show notes. There you will find links to the papers we reference, supplemental material, and a transcription. We hope to see you here next time on Experiment Exchange.